All right, welcome everybody to our lecture, kind of slash office hour, because remember that you are provided with all the lectures. So this space here is not really for me to lecture, okay? Because I've given you all that. I've all the information you need is there, but there are some concepts, especially this week, that I like, that are maybe a little more difficult, or maybe you'll definitely see. And so I want to spend just a little bit, some time on that, so that when you do get to it in lecture, it's going to be like, oh, okay, I've heard this already. Now you know exactly what was she saying again. So anyway, but I don't want anybody to think that, oh, I didn't cover that in the material this week, and that's okay because it's covered in those lectures. So let me just kind of go to the website, and just to make sure everybody's kind of getting in a flow, right? There's everybody's creating some kind of routine, maybe, to like how they're going to do this class, because it's pretty much the same every week. Just the material content changes, but what we do is the same every week. So last week's material was due on Sunday, and every Sunday everything is due for that week, and there is a penalty for lateness. Now, I didn't apply any penalties to last week's material, so if you didn't finish something last week, um, make sure you do it as soon as possible, and I will, get rid of any penalties from last week. Okay, as people are like trying to figure out, how do I do this class? But now from here on out, there is a 10% penalty for every day that it's late. So if you have to miss something, something comes up on Sunday and you didn't finish something, just turn it in as soon as you can. All right. And knowing that each day there'll be a little bit of a penalty. And in the long run, it's not going to hurt really. But points do add up. So and know that again, there you should be kind of setting up a routine. So every week there's going to be chapter stuff, right? So we cover, usually a couple chapters each week. And then for each chapter, there is a lecture to do and then a quiz. Those are two separate assignments that you need to complete for each chapter. And it's due during the week that it's posted. All right. So I would start early. Okay. Watch a lecture today, watch a lecture tomorrow, or, however you want to do it. And, but this week there's two lectures and two quizzes, right? Chapter 2 and chapter 3. So, just get in the habit of doing those weekly. And just want to remind people that these quizzes, they're taken from a word bank or question bank, right? So every time you take it, you're going to see a different set of questions. And I think I have it set to just keep your highest one. If it doesn't do that, let me know. I got to go back in and check my settings, but I think it's just unlimited, and you choose, it'll automatically put the highest score in, I think. If not, then that's not what I meant. Um, so I don't want anything to discourage you from doing it again and again, right? Um, so the more you take it, the more questions you're going to see. And it's from the same question banks that I do the exam, right? So if you just take it, get used to just taking the quizzes throughout the week. Okay, don't just think you need to take it once, take it multiple times. And that's like, even, you'd watch a lecture and just take a quiz really quickly to see how you do, and then just keep taking it. And then what will happen is, if you do that every week, then when it's time for the exam, you will have seen most of the questions. And then maybe you can go back and do these quizzes again for each chapter, just to practice. And then you're ready for the lecture quiz. So the lecture quiz shouldn't be this big, scary thing if you do the little things each week. Is that clear? And because what you don't want to do is just take it once, and then when it's time for the exam, then try to like take 5 lecture quizzes over and over and over again. Okay. I mean, you can, and you may want to do that, but that's just a lot more work, and it won't, you know, uh, be in that long-term memory if you're kind of trying to cram at the end. All right. So, but I'll leave that up to you, how many times you want to take it. I didn't really check to see how many times people took chapter 1 quiz, but um, and then we start something new. Okay, we're starting our bones. And every week there'll be a new set of bones. And this will go till I can't till March here, maybe. Bone 7, a week 7 bone list, and then oh no, to week 8. So March 22nd is the last bone list. And then this week before spring break is when you take your bone exam. Okay, so you're going to have an exam over all the bones. Now, since you guys are like my first class, you guys are the guinea pigs. And what I did is I decided not to have like graded weekly bone quizzes. We'll see how you guys do with that. And so what I have instead is because you want to be quizzed on your bones, right? You just don't want to study and then, wait until the exam. You want to have some mechanism to be able to study and quiz yourself. And so what I do have is ungraded bone quizzes. Okay, so just want to remind you, I think I mentioned it earlier, but there will be bone quizzes. And maybe in the future, I'll make these points, but right now there's no points to them. It's just practice. So we'll see how that works. If I see that people aren't taking it, then I may make it a required thing in the future. You know what I mean? So I just want to see how you guys do. But if you take these throughout the week, and they're like little short bone quizzes based on the clavicle and scapula. And again, these questions are taken from a question bank. And so for the bone exam, I'm going to use the same question bank. So the more you take it, the more questions you'll see. And so there's these practice quizzes that, or these quizzes and practice quizzes that I want you to take throughout the week. And this one doesn't have any consequences or whatever. It's not graded, but just the more you take it, the more, the better that bone quiz at the end will be. And it will help to, solidify things. Now, we do have a tutor, right? So make time in your schedule for tutoring. She's going to, I'll see what she does. She's been telling me things that she's going to do. She's going to work on these bones that are posted that week, as well as, going over stuff from the chapters. Okay. And helping you with that. I think she's making like little quizzes and things like that. I'm not quite sure, but she has stuff that she's planning to do in those office hours. All right. So it's not, just a question and answer thing. Certainly there will be part of that. But again, this is a new class, and I really don't think in the past the course had any tutors specifically for this class. So I hired a tutor just for this class, and she's very, very knowledgeable. Okay. Any questions? So, you've done the first week. Are there any questions as far as like, what do I do? Or because I do want to talk about some content stuff too. Questions? You guys good? Are you guys finding things okay? Yeah? All right. And if there are ever questions, remember that this is you guys are the maiden voyage of this class. And so if you find something that doesn't look right, then email me. Someone already found something. She was working ahead and she was on chapter 4. And when she clicked on the video, there weren't any questions in it. It was like just the video, no questions. And so she said, "Hey, I didn't find them, so I had to. I didn't link it properly, and I linked it to just the video and not the quiz." So anyway, things like that. Anybody who works ahead and finds a problem, I appreciate it. So let me know. So I fixed that, and now there are questions embedded. And so we're going to catch some of those little things. I want to remind you, if you were confused, like what are these videos? Okay, they're the same videos as the lecture, but these are the ones without the questions. Okay, it's the same thing. So if the questions are burdensome, like they keep popping up or whatever, and you don't want to deal with them anymore because you've already done the questions, and you just want to watch the video, this is where they are. So these are not anything extra. They're just the same thing without the questions, right? And then of course we have our images and then this practice quiz. So just start taking the practice phone quiz after you look at the material. Maybe go see the tutor and then start taking those practice quizzes. Okay. I really feel like these chapter quizzes also are practice. I mean, they are 5 points, but you can do it as many times as you want, and it's open notes. So I feel like, they're very low stakes. Okay. All right. You guys are good? Questions? Okay, so what I do want to do is talk about this week's chapters. Um, they're kind of involved. They deal with a lot of different concepts and ideas, and not super straightforward. So I would like to, just kind of talk about a few of them, but remember that this is not the lecture. This is like a review and things like that. And these actually will explain every one of the questions. All right, as you saw in the last week's. Okay, so let's go now to our overhead. And so we're doing chapters 2 and 3. So chapter 2 is, I'm just gonna say chemistry. And maybe that strikes fear in your heart. Um, I took a lot of chemistry, but I didn't really enjoy it a lot. Okay, but it's kind of a necessary thing to have an understanding of kind of the small things, that are kind of like the basis of living things. Okay, because we could just jump ahead to cells where kind of life begins, but there are things that are going to come up that if we didn't understand the smaller things, then it's just always going to be kind of like, oh, I didn't quite understand that, right? So we're going to go and talk about some of these little things. And I don't want to get too in depth, right? This isn't a chemistry class, but I want you to really just kind of understand, what holds things together? How are we held together instead of just kind of falling apart into individual little particles, right? And what forces allow us to do that? Because if we understand that, then, chemicals, drugs, different things, environmental things like radiation, and we're going to encounter these environmental things, and how will it affect, us being held together? And it can. So at the beginning, it asks about defining certain things like matter, mass, weight, an element, and an atom. So I kind of want to just start at an atom. Remember, an atom is just a tiny particle. And so every atom will have a what we call an atomic nucleus in the middle, and then little particles around it. Okay, so if we think of it, here's a small particle called an atom, and we have different atoms. Okay, there are carbon atoms and oxygen atoms and hydrogen atoms. All right, those are the different elements. So if we look at a periodic table in chemistry, they have all the different what we call elements, but they're really made up of individual atoms. Okay, and each atom or each element on that periodic table will have different properties. Okay, different characteristics. That's what makes them different. So how do they differ in characteristics? It's these little, what we call subatomic particles. And I think I mentioned protons, neutrons, electrons. So we have the protons in the nucleus, in the center of the atom, and those are positively charged. And then we also have neutrons, which do not have a charge. They are neutral in charge. That's why they're called neutrons. Okay, so they don't have a positive or a negative. Then there are little particles called electrons, which are negatively charged. And those are on the outside of the atom. Okay. Now, how, one like an oxygen atom differs from a hydrogen atom. It's the number of protons, neutrons, and electrons. Okay. And depending on how many, it creates different characteristics. So when I look at this, I see positive on the inside and negatives on the outside. And these electrons move around the atomic nucleus, right? So if I were to label this atom, this is the atomic nucleus, right? And I'm going to distinguish that from a cell nucleus. All right. Oh, colors don't show up very well. Okay. I just made this red, but so, an atomic nucleus is a small particle, but in the center that has the protons and the neutrons. Okay, so actually not all atoms have neutrons. The hydrogen doesn't, but anyway, that's kind of an exception. And typically the number of protons is the same as the number of electrons. Okay, so like a sodium atom will have 11 protons, 11 neutrons, 11 electrons. Okay. So overall, that sodium does not have a charge on it, okay, because there's the same amount of protons as electrons, same amount of positive and negative, so they cancel each other out. Now, one of the things that you may look at is what keeps the electron on the in this atom, because if it's floating around here, what prevents the electrons from leaving? Okay, it's this positive and negative attraction. So I think of it like, an orbit. Let's say the moon orbits the Earth, right? It's moving. What keeps it from floating away into space? There's a pull to it, right? There's gravity, there's gravitational pull that pulls and keeps that the moon in orbit around us. Okay, what keeps us in an orbit around the sun? Okay, it's again a pull that's keeping us in. So this is the same thing. The electron moves around an orbit, and what keeps it there? It's the protons. Okay, the positive charges in the center. So think it that way. But the more electrons that we get, okay, sometimes we have electrons out here. The farther the electrons, the easier they are to float away. Does that make sense? And so each atom has a kind of a stable formation, okay, of electrons and protons that make it stable. But when some atoms have electrons that are way outside, but by themselves, okay, then they're easily lost. So if we lose an electron, if an atom loses an electron, what happens? We have more positives than negatives. And in fact, in sodium, there's a lone electron on the outside. And so sodium will lose that electron. And so the overall sodium atom now becomes overall charged. And that's an ion. So an ion is an atom that has gained or lost an electron. Oh, let me move this. Okay, so we have these electrons, right? So when it loses an electron, it has an overall charge. Now it's called an ion. So I'll call this a sodium ion. So Na equals sodium. And you would think it would be like a SO or something, but that's in English, right? So natrium is sodium. It means sodium in Latin or Greek. And so, it just, that's why a lot of times it will have the same letters, like carbon is C and nitrogen is N, right? But sodium is kind of one of those weird ones. Anyway, so this up here is a sodium ion. Okay. Now, there happens to be another atom. Okay. So this one has 11 protons, 11 neutrons, and 11 electrons. That is sodium atom. A chlorine atom will have 17 protons, 17 neutrons, and then 17 electrons. Okay, that's what makes it a chlorine. Now, the thing about the chlorine is the configurations of the electrons. I'm not going to show you. You can look at it in your book and in the lecture. The electron configuration is such that there's an empty spot for an electron on the sodium. I mean, on the chlorine. So when they're together, one of the electrons on the sodium just likes to jump and fill up the electron spaces in the chlorine. And so if an electron jumps ship and makes its way to another atom, this one is going to be a chlorine ion, but now it's negatively charged because it gained an electron, right? And so now chlorine is negatively charged, sodium is positively charged. Now they're attracted to each other. Okay, and now they're going to be held together as sodium chloride. And notice the charges cancel each other, so there's no overall charge. And this is table salt. This is just salt. Okay, but there's different forces that are going to hold atoms together. And so what you'll learn in the lecture is some different, what we call bonds. So bonds are connections between atoms. Okay, and there's different ways that atoms are held together. I just showed you one way. One type of bond called an ionic bond. So it's a bond between ions. But just realize that that there's all these kinds of forces that hold atoms together to form molecules, right? So two or more atoms equals molecules. Okay, and so now we're building things, and now we made some molecules. And then, we put molecules together and we form structures and etc. All right. So I thought I'd just kind of explain this, what is an atom? And you kind of see that, we do have to go back and talk about some of these things. I'm not going to ask you a lot of details about it, but you do have to know, just know what I asked, the questions that I asked, the different types of bonds. But now you have an understanding of what holds us together, our atoms together, right? So if you have some kind of alien ray gun that disrupts the connections between atoms, then we just explode into subatomic particles. Okay. So, all right. Anyway, but you can see that, anything that would disrupt these chemical bonds would be a problem, right? All right. So that is the first question in chapter 2. Um, talks about chemical reactions. All right. How do we build molecules and take apart molecules? Because in our body, we're constantly building and breaking down material. Okay. We break down as we digest food and then take those nutrients and then utilize that, the energy formed in the bonds as we break it apart. And then, but we'll get into that a little later. All right. Uh, we talk about things like pH, acids, bases, things like that. So that's kind of part of this chapter also, and why that's important. And then also we get into some molecules. Okay, so here we were building molecules, and then now we're going to look at some what we call biological molecules. Molecules. And so you can see I'm skipping things, but know that you just watch the lecture and it's going to answer all the questions, right? So we put atoms together to form molecules now. And I talk about 4 types. So 4 major types of molecules. These are the carbs, carbohydrates. And if I were to ask you, what is the chemical formula for a carbohydrate? Okay, you could like rack your brains and like, oh gosh, I don't know that. But let's look at that word. Okay, carbo. I mentioned that particular atom or element. So when I say element, I mean an element is made up of one atom. I mean a whole bunch of atoms that are all the same. Okay, so um, so what atom did I talk about? I talked about carbon. Okay, so carbon is an atom. And so there it is. What else is in these molecules? What does hydrate mean? Okay, I saw you mouth it, huh? What is it?
Water.
Water, right? So this is something you already know. Water is H2O. Okay, there it is. That is a carbohydrate. Now I'm going to put a little n, and n can be any number because carbohydrates, there's small ones and big ones. And so in this question in the lecture, I talk about the different types of carbohydrates. So if n equals, let's say 6, then the carbohydrate is C6H12O6. But it just means that the ratio of carbons, hydrogens, and oxygen is always the same. It's a 1 to 2 to 1. So if N is 5, it's C5H10O5, right? And so all carbohydrates will have the same number of carbons and oxygens and twice the amount of hydrogen. Does that make sense? Okay. So, but again, it's about like understanding what, what the words mean. Um, all right. And I talk about different types of carbohydrates and why they're important. Another biological molecule, and another term you might see is organic. So biological molecules are, things, molecules found in living things. Um, Organic refers to actually in chemistry, it's carbon containing. So it contains carbon. And so, we attribute the term organic to mean, natural or, no pesticides or whatever. But in biology, organic means carbon containing. And then, found in living things. So next we have lipids. Lipids are, these are the fats, oils, phospholipids, which I will mention a little bit, steroids. So it's a whole, steroids. Okay, it's a whole big group. And then in the lecture, I'll talk about specifically, I will focus on the phospholipid because that's one that maybe you've never heard of. But I do talk a little bit about lipids. The next biological molecule are proteins. Proteins are um, composed of amino acids. Amino acids are kind of the building blocks of proteins. An example would be like hormones, which we'll spend some time on later. Enzymes, which I'll discuss in the lecture. Okay, so these are a lot of functional things that do function in our body are made up of proteins. And then lastly, we have the nucleic acids. Now, this is one that maybe people have never heard of. So maybe I'll talk a little bit about this. Okay, nucleic acids, because, I'll talk about each one of these in the lecture, so don't worry, right? But these are, people have heard of before. There are two types of nucleic acids, DNA and RNA. DNA is the genetic material. Okay. Oops, sorry. It's found in our cells, right? The chromosomes are part of this DNA. When I say genetic material, I mean it's what's passed on from a parent to child. Okay. It's the DNA that contains genes and information in it that's passed on to offspring. Okay, so something pretty important. A lot of people think of it as very mysterious, right? RNA is, there's different types of RNA, but it's important in making proteins, protein synthesis. All right, so how do you make a protein? Actually, the process is amazing, and if we have time, we'll talk about it a little bit because that is in actually chapter 3. Yeah. Okay. So I talk about DNA and I actually draw it so it's less mysterious. Okay. So, pay attention to that. Um, you know, um, Trying to think of, how much you guys are going to deal with genetics and things like that. Probably depends on what kind of data, what kind of information you're looking at. Certainly there's lots of genetic disorders and things like that. So having a basic understanding of what is DNA, the molecule, the DNA, deoxyribonucleic acid, what does that look like? Okay, what is it made out of? Because you have to have that understanding to understand not only genetic diseases, but also um, cell division, right? So if I cut myself, oh, magically it heals. Okay, it fills in. What is that? Okay, that's cell division. We begin as a single cell, right? An egg and a sperm come together, they form a single cell. Then how do we get this? Okay, it's cell division. And so that is part of understanding DNA is how do we divide the DNA every time our cells divide, okay, which is a lot. And so it's really just kind of a basis of knowledge that's going to be important in truly understanding, different diseases and processes. All right, so pay attention to that. Um, all right, and then chapter 3. I do want to take a look at chapter 3 and look at some concepts in there. Okay, so chapter 3. Chapter 3 is our cell. Okay, and cellular processes. So it begins with, and we know that the cell is the basic unit of life. Okay. So it's important that we understand the cell because, that's what makes us up. And we have to know the, requirements of a cell, the functions of a cell, because then, we can apply that to a whole organism. But, sometimes we have to deal on a cellular level if we're, trying to prescribe drugs or, it's going to affect the individual cells. So after talking about kind of general parts of a cell, okay, and then we also have to know the organelles, the different organelles and their functions. Okay, and that's a pretty straightforward thing, right? This is the mitochondrion. We call it the powerhouse. It makes energy, right? Now, let's talk a little bit about that. How we make energy. I want to know if it's here or the following. Okay, we may do it at a later time, but the basic functions of each organelle. So that's straightforward. The lecture will talk about the membrane and go into detail about the parts of the membrane, and we're going to, draw it and look closely because the membrane is what is going to interact with the outside world. Okay, so it's important to understand what it looks like. And I kind of want to jump ahead to looking at how molecules move around. Okay. Um, so pay attention to the membrane also. That's really important. So now we're going to discuss a little bit about how material can move around the body and also move into and out of cells. Okay, because we ingest things, we eat things, we drink things that are going to interact with our cells, and we have to be able to control what enters and exits our cells. Okay, and the cell membrane, that's what the cell membrane is so important. Okay, so here is our basic cell. We have a membrane. We have a nucleus. And everything between the membrane and the nucleus is called the cytoplasm. Cyto, C-Y-T-O, or cyte, means cell. So every time you see that term, it means cell. So if I say, what is a osteocyte? Okay, you just see the term cyte and you know that it's a cell. But what kind of cell? Osteo. Oh, that's bone. Okay, that's a bone cell. All right, myocyte. Okay, you see site, that's cell. Myo's like muscle. It's a muscle cell. All right. And so just kind of one of those things that's going to appear over and over again. And you just determine what kind of cell it is based on, what's preceding the term site. All right. So there's different ways that material moves around the body, some principles that govern movement of molecules. So I'm going to call this movement of molecules. And I want to stress this just because it's something that you're going to see. So the first type of movement are called passive, meaning it doesn't take energy. Okay, so these are, I'm going to put passive movement, meaning again, there's no energy required. It's like sitting in an inner tube in a stream. If you just don't expend any energy, you just float downstream. That's like these processes, they just happen without any efforts. The first type is called diffusion. So diffusion is the movement of molecules from high to lower concentration. Okay. I'm going to say until equally distributed. If it's like in a closed space. All right. So if someone came into a room and they had put some perfume on. Okay. Who is going to smell it first? Okay, so here's someone. They're going to put some perfume or cologne on. All right. That the molecule will be right on the person, right? You have a whole bunch of perfume molecules all over the person. According to diffusion, who's going to smell it first? Here's Joe. Here's Sally. Okay. Who's going to smell it first? It's this person. Let's say
The tallest one.
yeah. Why? Okay, I think people know, but it is diffusion. Molecules will move from where there's a high concentration, and they just begin moving. So that you just recognize that this is a principle that governs movement of molecules. Everything follows this principle. It just starts moving. And you can think of it like molecules, if they're a whole bunch tightly, all together, very dense amount, they're going to hit each other. And when they hit each other, they move apart. But if you don't have a high concentration, there's not many molecules, okay, they're not going to hit each other as fast. And so they're going to more slowly move. So if you have a whole bunch around you, they're going to just start moving. And so this person is going to smell it first, and then this person, and etc. It's just because molecules are actually moving and binding to receptors in your nasal cavity or, in your mouth. Okay. I used to talk about like a fart. Same principle, but it's disgusting. So, but hey, same principle. Okay. Molecules are moving from when there's a high concentration to where there's lower. And if they're in a room, eventually those molecules will equally distribute in the room. Okay. So, it's spread out in the room all over. That is diffusion. Let's talk about a second. I'm going to put it over here because I'm going to draw some things. A second type of passive movement is called osmosis. Osmosis is a special type of diffusion, but it's more specific. It's the movement of water. Okay, so it's very specific. Movement of water from high to lower concentration of water, lower concentration. And a second thing is it's going to pass across a membrane. Now, one thing about membranes is that they are what we call selectively permeable, meaning membranes allow some things to pass across and prevents other things from passing across. We call it selectively permeable. Selectively permeable means that, again, it's selective in what it allows to pass across, but water gets a free pass. Okay, so let's see how this applies to the medical field. Okay, so remember that water will go from where there's a lot of water to where there's a little water. Let me define a couple things. A solute is substance dissolved in a solvent. Now, the best solvent is water. Okay, so anything that you dissolve in water, it could be salt, it could be sugar, right? Something you dissolve in the water is a solute. A solvent is fluid that solutes are dissolved in. It's kind of going around in circles. So the best one is water. Okay, I shouldn't have put this here. I put solvent examples water. So the best example of a solute is like salt or sugar. Okay, those are good solutes. Good solvent is water. All right, so now if we take salt and we dissolve it in water, let's say I put it in a beaker here, and I put some water in here. Let's say I put 100 milliliters of water out here, and I dissolve salt. So let's say salt. Let's say 10 grams of salt, NaCl. This is I'm not going to ask you this. This is a 10% salt solution, NaCl solution. Okay. But what I want you to see is that there's a salt component and there's a water component. Now, if I add 5 grams of salt, less salt, we're going to get a smaller 5 grams in 100 milliliters of water. Okay, that's going to be a 5% salt solution. All right, so what we have here is where is there more salt and where is there a higher concentration of water? Okay, so different way to think about this. So here there's more salt. Okay, and so the concentration of water is less. If this is 5 grams of salt, less salt, but the concentration of water is more. Okay, because there's less salt in it, so there's more water. Okay, you could look at it like, you know, if I were to measure it, here's 10 grams of salt here, and the rest is water. And this is This one is 5 grams of salt, and the rest is water. So this is water, and this is salt. All right, so do you see? You can see that, oh, there's more water here than there. One way to remember when you're figuring this out is water always follows the solute. Okay, so this is something to remember. So now let me put it in relation to something useful. Okay. Might you read something about an IV, someone given an IV? Okay, there's different amounts of water and different amounts of solutes in that solution. Okay, and there's you know you can get different solutions. So, and you put the IVs into your veins where you have red blood cells. So if you have a red blood cell here, okay, and a red blood cell, it's what we call it's like it's shaped like a donut without the center completely cut out, right? So it's thinner in the middle than on the outside edge. So this is a red blood cell. And inside red blood cells, there is a known salt concentration. So inside, we have a little bit of salt in there. It's less than 1% salt. So it's a little salty on the inside of cells. All right. It's actually 0.9% NaCl. Now, if we surround that red blood cell with an IV, with fluid, is it important what the solution is that we surround it with? Is it important what, can we just give somebody water? Okay, let's look. If we give somebody 0% NaCl, okay, that's how much salt, 0 salt, it's actually 100% water, all water, no salt. Now remember what I said, water is going to always follow the solute. Now the thing about membranes is salt is too big to pass across. It has to have special mechanisms to do it. However, water will go. So our only concern here is where does water flow? Because the salt doesn't go anywhere. And remember that the water follows the salt or the sugar, whatever it is. Okay. All right. So where is there more water? And it's going to go toward where there's less water. In other words, the water is going to go toward where there's more salt. So in all these cases, you have to just identify where is more salt. Where is there more solute? And that's where water flows. So based on this, oh, there's a question. Okay. Oh, Pearl left. Okay. So in this scenario, where will water flow? Okay, so you have to look at where's there more salt. Wherever there's more salt, that's where the water goes. 0 salt, less than 1% salt. The water's going to go toward where there's more salt. Did you figure this in? The water's going to go into the red blood cell, the cells. And what's going to happen to that red blood cell? That red blood cell is going to go whoop, and it even might burst. This process is called lysing. To lyse is to burst. This solution on the outside has a name. It's called a hypotonic solution. Hypo means less. So if you die from hypothermia, you went up in the mountains and you got separated from your party and you had to sit in the snow all night, okay, you might die from hypothermia. Thermia means temperature, hypo means less. Your body temperature is going to drop. Okay. A hypodermic needle, derm means skin. Hypodermic is a needle that's going to go under the skin. Okay, so you're going to see hypo a lot. All right, what if we put our red blood cell into another solution of a 5% NaCl? And remember, inside is less than 1%. So the question is, where does water flow? Water will move, right? All you do is figure out where's there more salt? Where's there more solute? 5%, less than 1%. Oh, there's more salt out here. I think of it like the water goes to try to dilute the salt or the sugar, right? So in this case, water is going to move out of the cell. The red blood cell is going to shrivel or crenate. Crenate is to shrivel. This solution is called a hypertonic. Hyper means more, right? If you're hyperactive, you have more energy than the average person, okay? So this has more solute, okay? So what kind of solution do we want exposed to our red blood cells? What should be in the IVs? In which type of solution will the red blood cells be happy? A 0.9% NaCl. Okay, so when I say less than 1%, less than 1%. Okay, so I should have put 0.9 because that's the actual percentage. All right, why? Because this is an isotonic solution. Iso means the same. So what's going to happen to the water? It's not going to go in, it's not going to go out. It will maintain its shape and its integrity. So this is a happy red blood cell. Okay. We'll imagine it being happy. All right, so okay, that's important. The percentage of the fluid that goes into your IVs. Should you take water and inject it into your veins or into your body? No, because your cells have different varying amounts of solutes, and so you want to match it. If there's surgery and someone's doing a biopsy on a piece of tissue like a liver, hands it to you and says, keep it moist, take it to the lab. Okay? They just cut something off, a piece of liver off of somebody. They're going to sew them back up, and they have this one piece of liver. Are you going to put some water on it? No, because water is a hypotonic solution. The water is going to flow into the cells. Now, how does this apply to like other cells, like some lettuce? Okay, so here's a piece of lettuce. And you take it home. It's a little wilted because it's been just sitting, desiccating, okay, in the air. And so you wash it. So if you put water on it, a hypotonic solution, 0% salt or sugar. All right, what's going to happen to the cells? The water is going to move into the cell and it's going to cause it to plump up. or it's going to get crisp, nice and crunchy. Now you take that same lettuce, now it's all crispy, and you add dressing on it. What is dressing? It's a very hypertonic solution. It's got salt, it's got sugar, all right, lots of solutes. Where's the water going to flow? Into or out of the cells? Remember, water flows toward where there's more solute. More solute out here. And what is that lettuce going to be like? It's going to be soggy after a while. It's all osmosis because water will move. And notice after a while, you're inside your bowl, it's watery, right? The dressing is watery. Why? Because it's just sucking the water directly out of the lettuce. All right. And so that's what happens in our body too. And all our fluids and everything are all isotonically, isotonic. Let's think about diarrhea. Okay. So, okay, when might you have diarrhea? Okay, there's different reasons, but let's look at like, sugar-free candy. Anybody ever eat a lot of sugar-free candy? What happens? You get diarrhea. Why? Osmosis. So in sugar-free candy, the sugar is a sugar you can't digest. Okay, it's, I don't know, one of those I don't know, allulose or xylulose or, all right, the sugar you don't break down. So, but it's still sweet. So it still has some taste to it. But the reason it doesn't, there's no calories in it is because you don't have enzymes to break the bonds down. So it stays intact as sugar. Okay. Fake sugar. I'm going to call it fake sugar. It doesn't break down. So it goes through your digestive system into your colon. So if this is your colon, your large intestine, and now you have sugar in there. Ooh. Okay. So not only are the bacteria happy, so you have bacteria, but also it creates osmosis. So this is sugar, where water is going to flow into the gut because of all that sugar in there. This is if you eat quite a bit. And so what happens? Then you get diarrhea. Diarrhea. Okay. Does that make sense? Okay, it's all about osmosis. Osmosis affects so many parts of our lives and health. And so it's important that you have a good understanding, in the medical field. When you administer drugs, you have to think about things like that, osmosis. Okay, so I talk about other different types of movement of molecules, and but that one takes a little bit of thought. Okay, and then it ends with a couple things that I didn't get to. It's about, going back to the DNA and how do you make a protein? And so this is involving DNA and RNA and how those work to produce a protein. And so I like to think about it like, if I start working out, okay, and then my muscles will get bigger, right? Depending on, what type of workout. But if you're looking to bulk up, right, you start lifting weights, you have to make specific proteins, the muscle proteins, to fill in the muscle cells, the myocytes, right? And so how do you make that particular protein? Okay. And the code is in the DNA. The gene for that protein is in the DNA. And how do you get from the DNA, the gene in the DNA that's going to tell us to make that particular protein? So that is part of the lecture, is It's showing you that process. And you know that process is really important because we're going to make all kinds of proteins in our body, right? And so, you know, that's how we differ, right? Our DNA differs a little bit. And so, you know, the gene is a little different between you and me. And so, you know, we might make different proteins of the same, you know, the same protein, but it might vary. And I'm thinking like eye color or hair color, right? We need to make certain proteins that aid in who we are as a person. All right, and then it ends with cell division and division of the nucleus and the DNA. So that's called mitosis. So there is a lot. So I would start early on these lectures because they're kind of long and really content dense. There's just a lot. As you can see, there's a lot of concepts that are difficult. And so, you know, start it early, take practice quizzes, see the tutor, either so the tutor is Wednesday night. Let's finish with that, just to double check. Okay, so the tutor, let's go to the tutoring. It's in the welcome. Go to the tutor schedule. Okay, and Wednesday night, 6 to 7:30, and then Saturday morning. All right, so go to see the tutor, and she can, you know, explain any concepts, but you know, you need to have looked at the material first and before asking questions. But then she'll also work on the bones. Okay, and that really is their specialty, is the anatomy. And okay, let's see. That was a lot today. And again, don't wait to the last minute because it's pretty dense. Okay, you guys. Oh, okay. If someone had too high red blood cell, instead of the salt, because it's the, you know, it doesn't change the amount of red blood cells, the salt content will only influence maybe the shape of the cells or the red blood cells themselves. You'll want to look at the process of making red blood cells. Okay, so thrombocytopenia, um, or that's too low, right? You can give a hormone to produce more red blood cells. If you have too many, they have to first figure out why you have too many. Um, and we'll talk a little bit about like blood doping and things like that when we get to the cardiovascular system. But having too many red blood cells is a problem also because it can clog small vessels, capillaries, and cause like maybe strokes and things like that. So you would want to look at the reason why you have too many red blood cells and then work from there. But red blood cell production is controlled by one thing is by hormones called erythropoietin, secreted by the kidneys. So, okay. Any questions? Any questions before I stop the recording? All right, you guys. Thanks for coming.
Thank you.
Hang out. Email me with questions or problems, concerns. If you see a problem, then let me know. But go see the tutor. That would be very helpful.
Okay, thank you.
Okay.